again would like to thank all the members of the panel. This has been a very enlightening time for those of us on the committee and in the audience for the things that you've been telling us. Of course, there's lots of information that's there. So let's open this up if there are questions, additional questions from members of the committee.
Well, I have just a simple one for Marianna in the last presentation. In terms of the adverse outcomes pathways charting, what has been done with nicotine in that regard? And because you had your comparisons for most of your other in vitro tests were with a tobacco smoke condensate or a standardized extract of some sort. What role does nicotine itself pharmacologically play in any of those? Probably not in any of those other assays, but I'm interested in looking at the pharmacology of nicotine and from an in vitro perspective and what might be learned from adverse outcome pathways.
I believe from the adverse outcome pathways program and on AOP Wiki, there are a number of different AOPs that have been put up there to share with the wider community around those particular aspects with nicotine, but not from a perspective from e-cigarettes, but more from developmental and reproductive toxicity.
I would like to ask the whole panel the two questions. The one would be, we probably will need to come out with some general statements about the ends product. And also we would like to consider differences across the products and how different product characteristics may affect the outcomes that you observed in the studies, or they might affect the users of the different types of products with the different flavorings, different PG-VG ratio, etc. What would be your comments? Like, what are the general statements, general conclusions from all the studies versus when we should be very careful when we consider different products?
I'll start as a general position, and we have scientists here who can get down into the nitty-gritty. But one of the things that's not clearly understood, in my opinion, about these products is that, much like a recipe, you change one ingredient, you change the final dish. Like in any engineering situation, if you take out one part and replace it with something else with a different specification, you may very well change how that device operates. It is very much the same here. It's not consistent. If you create a reference device, it's ultimately going to be referenceable only to itself. Because if you change a component, then you change. I mean, one of the things I didn't have time to get to in my slide that I think is really, really relevant in these discussions and these evaluations is that when we get to smoking cessation efficacy evaluations, right now the direction is a paired liquid and device, very specific. But that's not representative of the real world. In the real world, people have hundreds of device combinations with different tanks, with different devices, with different liquids, and those all yield subjectively satisfactory outcomes. And so that subjective satisfaction, it plays a key role in the cessation efficacy. And if you only deal with one of the things I talked about was the first-time vapor often will try 20 or 30 different liquids, usually looking for a tobacco that's going to similarly replicate what they're giving up in tobacco smoke. Once they stay with vaping for 2 weeks, if they're not dual using, as long as they're not recoding the olfactory and taste senses with tar, then those senses restore. And many people find that the tobacco flavors are no longer pleasant and enjoyable or subjectively satisfactory. So they start all over again with different ones. So the airflow, the power levels, some atomizers produce what we call, some people call flavor chasing as opposed to cloud chasing. There are a lot of different variables in here, and each one yields a different character of the vapor. It could be realized in warmth of the vapor, thickness of the vapor, the amount of the vapor, the throat hit, which might be a mild irritation or expansion feeling. It is very, very relevant, and I think we have to be really careful when we look at science to assume that any one particular combination is going to be representative of anything beyond itself.
I'm going to respectfully disagree with Lou. So if you say we are at the point where if you change any one thing, everything gets thrown up in the air and who knows, that's not engineering, that's tinkering. So as an industry, to be an engineered discipline, we need to get to the point which... we're making progress towards, but certainly we're not quite there, and I don't know that anybody is, where you say, well, I'm going to change something because I want a specific result rather than let me change something and see what happens. So I think really in order for us to be able to make generalizable statements about the industry, the goal is make it an engineering discipline. Nobody says, well, let's build a bridge and, maybe it'll sit up, maybe it won't. Let's run a couple thousand cars over it and, if it looks good, we're good. Because we have engineering disciplines, we say we can calculate the stresses and the strains and the wind loads and the resonant frequencies and whatnot, and the bridges don't fall down. Every so once in a while, you get a Tacoma Narrows, but for the most part, you build a bridge, you get a bridge. So I think really as an engineering discipline, we need to get to the point where you say, okay, I want this temperature, I want this distribution of particle size, I want this amount of dosing, I want, so we can be designing products to specifications rather than sort of, throw it together and hope, which is always where, engineered products start. But, we need to very rapidly, and I'm hopeful that PMTA process is sort of shoving people that direction. Hopefully we can get to the point where you say, I'm going to know with pretty good certainty what we're going to get before we make a product rather than after we've characterized 50,000 of them.
I would clarify that that's wonderful in terms of looking forward to new products. My response was more of an observer, looking at what we have today, and I have complete respect for John, and he's 100% correct.
If I could jump in, I think we need to respect where the science has gone with this product, if we could say a product, from consumer demands. This is not a pharmaceutical product that went through all sorts of stuff to be able to get on the market before the consumers actually get to it. The consumers demanded higher power with lower decomposition. They demanded it. So the engineers created it. Okay? We're kind of coming in late on. Oh, what does this do? What does this do? Can we quantitate this? But I think we have to respect that this is an industry that has been driven by consumer demand and consumer discernment. And so that's all I wanted to say about that.
If I just jump in, from a testing perspective, if you look at, say, the framework I put up, and there's many other frameworks, and you look at PMTA, MRTP, the resources and effort required to actually substantiate reduced risk nature of e-cigarettes is profound. It's tens of millions of dollars, and it's years of research. So I'm not saying that doesn't have to be done, but I think what needs to be done then is to create some sort of strategy where we can actually then establish a foundation data set and then bridge to subsequent variants. And using all the expertise we heard about today, we're getting more and more understanding around the sort of operational envelope of the product, around chemistry, in vitro, clinical endpoints. So we need to sort of leverage that SME expertise that exists, establish the foundational data set, and then pragmatically bridge to different product variants.
Mm-hmm. And actually, my second question is to James and Marianna. You mentioned at the very end of your presentation the link between your in vitro studies and clinical outcomes, and this is probably when we really need the answer. And you showed different models with the different mechanisms, and when we're talking about a reduction and the gap, the reduction in the risk from between vaping and smoking, we consider different clinical outcomes, whether this will be respiratory disease, whether it will be cancer, whether there will be cardiovascular, and the others. Based on your in vitro models, when do you expect that the clinical difference is the biggest and there is a huge benefit, when it might be, the gap may be not so huge? I mean, the difference between vaping and smoking. I don't know if it even makes sense.
Kind of, yeah.
Yeah, so let's say cancer risk versus cardiovascular risk.
Yeah.
Based on your in vitro models, are you expecting the same reduction of I don't want to put any number, but
This is kind of like a very novel area around trying to test the ends within vitro. And again, with the advent of 21st century toxicology and looking at some of the new omics platforms that are out there, really starting to build up the mechanistic data, we can then take this to help us start to define better prediction models around, these ends models. And going back to James's point, if we can have a fundamental data set already on a basic product and then understand what the product changes are over a period of time, we can then start to build up that knowledge base and the predictive power of our in vitro models. What really lends itself in particular are these 3D model systems that are becoming more available to us in organs on a chip. These are starting to look more physiologically relevant to what you would see in an in vivo scenario. And again, if we can start to look at clinical models and take the tissues available to us from clinical studies and relay them back into the in vitro scenario and build up these compare and contrast activities, which we have done in the past with our serum studies from our smokers and done very, very similar studies in vitro using these 3D systems and collected the artificial surface liquid and done the comparisons. We can really start to build up these predictive models to help give us that answer in the end.
Thank you for all of your talks. I think for Dr. Kurt Kistler, when you were talking about comparing e-cigarettes to air and then e-cigarettes to cigarettes, right? So you have like two extremes. And then I think you mentioned some other potential benchmarks, which might be in the middle. You talked about some occupational safety kind of thresholds. So general question for you and maybe the other presenters on this panel, what are some other good comparators that may not be cigarettes and may not be air and might be somewhere in between so we can gauge the relative health effects of e-cigarette products?
That's a great question, and I don't know. I just don't know. It's a matter of, I guess, coming up with a particular scenario and being able to quantitatively measuring it and then being able to translate that into breathing rate over the course of a day. And, the OSHA and the NIOSH levels were about air concentration and assuming a certain breathing pattern over a certain period of time, and then, with tidal volume and everything, and then translating that into an exposure in terms of an amount over the course of a day, and then taking that amount and saying, okay, how many milliliters does a person go through on average in a day? So then what does the concentration have to be in the e-liquid to be able to get under that exposure limit? But there's so many approximations in all of this. I would like to see, we use NIOSH limits to estimate back to e-liquids. I'd like to see that looked at closer so that we can even be able to judge as to whether our approximations, which no one has challenged, whether our approximations are actually correct. So I think our first step is to actually nail down these quantities that we know in terms of workplaces and more intelligently get back to what actual exposure could be expected. And then start to think about other scenarios, maybe vaping in a car, sitting next to someone else, that sort of thing. But, I think we need to be looking more deeply and accurately at quantities that are well known, and then be able to say, okay, well, now where do we go? So I hope that answered your question at all.
I think so, yeah. To a certain extent, yeah.
May I jump in just one additional comment
Sure.
there, because I really appreciate the the cigarette and the air comparators. We published a paper last year looking at over 100 toxicants on e-cigarettes, and we've been measuring toxicants in cigarettes for decades, and they're up here. Whenever you go to e-cigarettes, the levels are so much lower. You've got to be so careful about actually the residual air environment. So what we had to do for our publication was to measure the cigarette, to measure the ends product in separate labs, but also to measure the laboratory air, because there was actually residual levels of toxicants from the external environment, from the lab operators, and they were actually giving us quantifiable levels of toxicants. So you would have got a false reading if we hadn't have done those lab blank measurements. So just I think a good tip for people when they're doing the chemistry measurements is to look at things like that.
Yes.
I might just try to add to that. If you're talking about comparators, and I'm not sure I heard the clarification as to what you were looking to compare, environmental exposure or first-person exposure, but I would say if we're looking at the continuum of risk, then we should look at the continuum of risk across everything. So you have air at one end of the spectrum, you have smoking at the other end of the spectrum, and then you've got electronic cigarettes, NRTs, gums, patches, lozenges, and so forth, heat not burn products. I mean, if we're going to look at the continuum of risk, let's look at all of it and try to put things into perspective so we understand it.
This question is for Dr. Murphy. Dr. Murphy, you presented the results of a dynamic population model in that the model assumes 5% of the mortality risk compared to cigarettes. What's the basis for that assumption, and how much consensus is around that figure?
Okay. Okay, so just to start off, I mean, we've been doing population modelling now for about 2 years, so we're just building our expertise in this area, and that was, I think, our first publication in the area. So what we did was we took the Public Health England view on e-cigarettes, that they have the potential to be 95% reduced risk versus cigarettes. So that number came up from a Delphi panel that looked at over 180 scientific studies to come up with that number of 5%. Now, for me, what will be really interesting over the next sort of 12 to 18 months as we go through the studies on both electronic cigarettes and tobacco heating products will be to actually get our own excess risk estimates and then plug them into the model and see if we get the same result.
Yes, go ahead.
I just had a question for Marianna. I thought the in vitro approach that you described is very rigorous and could be very useful. I had one question though. You were using immortalized cell lines, like the Beast2B cell line, to look at your outcomes. Why isn't it that you're looking at primary cell lines that may actually respond a little bit differently?
Across all our in vitro test systems, we use a number of different cell lines, as you rightly said, but also we move into some of the primary tissues, for example, like the human bronchial epithelial cells, depending on the specific endpoint of choice, and also trying to take a number of these cells to the air-liquid interface. It really hinders on a lot of the technicalities and the suitability of that cell type of choice. So some of the assays that I indicated to you are just, from a routine perspective, make it easier for us to utilize some of the continuous cell lines, and also the endpoints that you get and the results are very, very similar to what you would see in primary cells. But moving forward, a lot of the tests that need to be done would be more so in the 3D tissue systems, and that's where we really need to be working in.
Okay, thank you.
But the 2D just allows you to have a very quick look-see to start to put you in the right direction.
So this has all been very enlightening, from in vitro to in vivo to experimental cultures. We really appreciate what you've talked about. It's interesting to me that on our final speaker we heard over 8,000 different formulations of e-cigarettes, and in our first speaker that we need to have standardization. And that will be a challenge for us as a committee to look over the literature as it exists. We'll take a break now.